Pyruvate dehydrogenase is a very large enzyme, and it's an enzyme that is used after glycolysis has occurred. Now remember, the products of glycolysis are two pyruvate molecules. Now those two pyruvate molecules in aerobic conditions will be converted into acetyl CoA, and then the acetyl CoA will be fed into the TCA cycle, or the Krebs cycle. So in order to go from pyruvate to acetyl CoA, we use the enzyme pyruvate dehydrogenase, and this is a very large enzyme. This enzyme has three major subunits. The first one referred to as the E1 subunit, or pyruvate dehydrogenase subunit. E2 is the dihydrolipoyl transacetylase subunit. And E3 is dihydrolipoyl dehydrogenase subunit. Now, if we talk about in terms of where these subunits are located, the dihydrolipoyl acetylase will be located more on the inside of the enzyme, and then pyruvate dehydrogenase and dihydrolipoyl dehydrogenase will be located more on the outer portion of the enzyme. Now, if we take a look over here in this depiction, we can actually see more clearly that we do have three subunits. We have E1, E2, and E3. And each subunit has a specific prosthetic group. And we'll talk about those prosthetic groups. And once again, to reiterate, these are just the names of each of the subunits found within the overall enzyme known as pyruvate dehydrogenase complex. All right, so let's look at the mechanism. So first, let's look at a general overview of the mechanism. So as I mentioned, we start off with pyruvate and we go towards acetyl CoA. So let's look at the molecule of pyruvate. We have 1, 2, 3 carbons in pyruvate. And then we also have this carboxylate group over here. So pyruvate is essentially an acid. But if we take a look at acetyl CoA, we have this coenzyme A group right over here, and we have 1, 2 carbons. So 1 carbon from going from pyruvate to acetyl CoA is going to be lost. And we can actually see that carbon over here, and it's lost as carbon dioxide. So now if we look at the enzyme, once again, the enzyme is the pyruvate dehydrogenase complex consisting of E1, E2, and E3. Then as I mentioned, each subunit of pyruvate dehydrogenase has a specific prosthetic group. Now we have 3 prosthetic molecules that are very tightly bound to pyruvate dehydrogenase. So the first prosthetic group is FAD, the second is lipoate, and the third is TPP. And we'll look at where each of these is found. And then lastly, we can see that we have NAD+, and in the process of going from pyruvate to acetyl CoA, NAD+ is going to be reduced into NADH, and we'll get that proton. Because remember, whenever we talk about the reduction of NADH, there's always that proton involved. All right, now let's go down and look at the details of this mechanism. So as I mentioned, we start off with pyruvate. Pyruvate is a 1, 2, 3 carbon molecule. We have this carbonyl over here, and then the end is a carboxylate. Now in the first step, what happens is that we have TPP. Now this over here represents the entire structure of TPP. And as we can see, TPP is a very large molecule. But what part of TPP are we specifically interested in? We're interested in this negative charge right over here. That negative charge is on a carbon. So that carbon is able to form a stable carbonyl anion. And that stable carbonion is going to act as a nucleophile, and it's going to attack the second carbon on pyruvate. All right. So one thing we need to know about carbon is that carbon can only have 4 bonds. Okay. So if TPP, also known as thymine pyrophosphate, if it's nucleophilically attacking that second carbon on pyruvate and it's trying to form a bond with carbon, carbon must break one of its bonds, because we can see that carbon has 2 bonds over here, 1 bond here, 1 bond here, for a total of 4 bonds. So it must break a bond in order to make a bond. So in order to make a bond with TPP, it's going to break its bond with the adjacent carbon, holding the 2 oxygens. So when we break that bond, we get a release of carbon dioxide, because see, we have 1 carbon, we have carbon 1, and then we have oxygen, and as a result, we get CO2. So when that's lost, TPP is able to form a bond with pyruvate, and as a result, we get this structure. We get TPP bound to what's left of pyruvate, and we're going to refer to this as the acetyl group. So the acetyl group remains, and it binds to TPP. Specifically, it's binding to this negative carbonion right over here. So this resulting structure of having TPP and having that acetyl group is referred to as hydroxyethyl TPP. So hydroxyethyl TPP. Sorry. Okay, this structure over here. Now, this step of TPP binding with this acetyl group and removing that carbon, forming carbon dioxide, this is all happening on the first subunit. So if we go back up here, this was our E1, right? On our first subunit, we had the prosthetic group TPP, and TPP was able to nucleophilically attack pyruvate and form a bond with pyruvate in the first subunit of the overall enzyme pyruvate dehydrogenase. And then we got the resulting structure of hydroxyethyl TPP. Now we're going to take a look at the second subunit of pyruvate dehydrogenase. In the second subunit, we have lipoate. Now, lipoate consists of lipoic acid, which is the structure right here, and lipoic acid is bound to a lysine. So in this diagram, we are depicting simply lipoic acid, and we're just writing lysine in the middle. So lipoic acid has this sulfur bond. So we have these 2 sulfurs binding to one another, and we refer to this as these sulfurs being in their oxidized state. Okay. When TPP binds with the acetyl group, lysine, remember lysine is a part of the second subunit, so it's found in the middle of pyruvate dehydrogenase enzyme. And it's in the middle, and it swings its arms, and it notices that TPP has acetyl bound to it. So it picks it up. And when you can see, when it picks it up right over here, it forms a bond with the acetyl group. And we can see that bond over here. So it forms a bond with the sulfur. Now we have sulfur plus an acetyl group. And in this process, we refer to lipoate being reduced. We can see the reduction happening right over here. And this sulfur forms that bond with that carbon. So lipoate is now reduced. Now this sets up the stage for the next reaction. And that next reaction involves a very large molecule known as coenzyme A, right over here. So coenzyme A is a very large molecule, but we are going to focus on this part of coenzyme A. And this is the name for the specific part of coenzyme A that we're looking at. So if we look at the specific part of coenzyme A, we see this sulfur in its reduced state, and it has that hydrogen bound to it. This sulfur is a very good nucleophile, and it's going to nucleophilically attack the carbon bound to the sulfur on lipoic acid. Okay? So it attacks, remember, carbon can only have 4 bonds, so it has to break one of its bonds, and it's going to break the bond that it has with sulfur. When it breaks that bond, as a result, we get the acetyl group bound to coenzyme A, specifically this part of coenzyme A, and as a result, we get acetyl CoA. Acetyl refers to, once again, simply this part, and now coenzyme A is bound. And then we get the product that we're looking for. We're looking for acetyl CoA. Now, I want to reiterate that coenzyme A is a very large molecule. We are specifically just zooming in and only looking at this part of coenzyme A, the part that's undergoing the reaction. So when we get our product, so we see coenzyme A coming in, and we get our final product of acetyl CoA. And now we have to think back and remember, whenever we use enzymes, we want to regenerate that enzyme because we don't want to alter the enzyme. We don't want to use it up. We have to regenerate it. It has to become what it exactly was in the beginning. And we can see over here that lipoate is in its reduced state. It's in its reduced state, and we have to oxidize it so that it can be ready to pick up another acetyl group. How are we going to oxidize it? We have to oxidize it through an electron carrier, and that electron carrier is known as FAD. And FAD is found on the 3rd subunit of the pyruvate dehydrogenase enzyme. So if we go back and we look at our enzyme depiction, we had TPP over here, we had lipoate on this one, and lipoate consisted of lipoic acid and lysine. And then we have on E3, we have FAD. So now we're looking at E3, and on E3 we have FAD. And FAD picks up the 2 electrons, and it gets reduced into FADH2. But then FADH2 passes those electrons onto NAD+, and NAD+ becomes NADH and H+. Now we might have the question, why are we using 2 electron carriers? Why couldn't we just directly transfer these 2 electrons onto NAD+? Well, that's because FAD has the ability to pick up 1 electron, or it can pick up 2 electrons. Whereas, FAD has the ability to pick up 1 electron or 2 electron, whereas NAD+ has the ability to pick up only 2 electrons at a time. So this lipoate, it's going to pass on 1 electron, FAD will take it, and then it's going to pass on the other electron, and FAD will take it again. NAD is not capable of accepting 1 electron at a time, so that's why we don't use NAD+. So once FAD picks up both of these electrons, then it can directly pass on both of the electrons at the same time. Okay, and then we get NADH and then H+. Now, if we look at the overall reaction now, starting from the beginning, we start off with pyruvate. TPP, the stable carboanion on TPP, will nucleophilically attack that second carbon, and we get this hydroxyethyl TPP. And then we have lipoic acid, which is swinging its arm around in the center of pyruvate dehydrogenase, and it picks up the acetyl group from TPP, and that essentially it acts as this transporter to take it to coenzyme A, and the coenzyme A reaction happens because we get that nucleophilic attack once again, and we get our final product of acetyl CoA. And then the last part is just regenerating oxidized lipoate. So we use the electron carrier FAD, and we get FADH2, and then we pass on those electrons to NAD+, to gain NADH. So overall in this reaction, we did gain 1 NADH, and H plus, and then we gained acetyl CoA, and then we also produced carbon dioxide gas. Now to quickly just go over the names. So remember, pyruvate dehydrogenase, it's a very large enzyme. You can see over here. It's a very large enzyme. On the exterior, we're going to find E1. On the inside, we're going to find E2 because E2 is that, it contains lipoate. Lipoate's that arm that's going to swing everything around to get where it needs to go. And then on the outsides, we'll find E3 as well. So these subunits, once again, they have names. So the first one, E1, is pyruvate dehydrogenase, E2 is dihydrolipol transacetylase, and E3 is dihydrolipol dehydrogenase. And all together, this forms the pyruvate dehydrogenase enzyme complex, which catalyzes the reaction from pyruvate, which are the products from glycolysis, into acetyl CoA, which will be fed into the TCA cycle in aerobic conditions.